Okay, so you guys are going to have to help me a little bit. Okay, so when I leave things out, because I'm doing this all from here, if I leave anything out and like there's something that was on your list of things to study that I don't mention, because my goal here is to put everything on the board in a place that makes sense. Okay, we'll see if it works. So you'll have to say, hey, you didn't say blah, blah, blah. And they'll probably go, huh? But we'll work it out. So when people come and ask me how to study, I usually say organize it into some kind of logical grouping system or whatever. And so people are coming and they're saying, well, so I've organized all the pathways like this. I'm like, I don't want you to think of it as I need to memorize these pathways. I want you to think of it as you need to understand different mechanisms and use the pathways as examples. Because the purpose of understanding pathways is because one day you'll be studying a pathway, or not. But let's say you'll all be scientists. You'll be studying a pathway, and some mechanism will be, you'll be studying it. You won't know it, and you'll have to come up with it from somewhere. And usually the way you hypothesize things is from your prior knowledge. And so I'm trying to give you this toolbox that you can use to think about signaling. Okay? And so we're going to start at the top. And by the top, I mean outside of the cell. And so I'll start over here. So outside of the cell, so we're talking about signaling. So what's outside of the cell? Someone raise your hand because that was a whole lot of words. Yeah. No.
I was going to say extracellular membrane.
Okay, well, no. In terms of signaling, extracellular, yeah. Some signaling molecules. Okay, so that'll be our first category. Okay, what kind of signaling molecules are there? I gave you categories. Hydrophobic. Okay. What else? Hydrophilic. And what else? What other signaling molecules, like when you talked about Notch and delta? Another receptor, right? Okay, receptor on another cell. Yes. Yep, I'll try to say everything because I know that you're not all going to equally see the board. So raise your hand if you can't see something or you miss something. Okay, so those are our signaling molecules. And they're outside of the cell. So let's make our plasma membrane. Okay. So when I say hydrophobic signaling molecule, what are you thinking about? What does it do? Yes.
I thought you were asking hormones.
Hormones diffusing across membranes. Okay, so hormones, steroids. Yeah, that's like the difference between my hand and my brain. I said steroids and I wrote stroids. That's okay. Steroids and diffuse across membrane. Across membrane. Okay, so let's have it diffusing across membrane. Okay, and then we talked also about nitric oxide, right? That also will diffuse. So we'll put that in that category of things that diffuse across membrane. And so maybe under that we'll do things that diffuse. We're going to make a slash diffuse across. Okay, and then, okay, so that's one category under that category. Okay, so hydrophilic. What do you think of? You don't need to give me names of things, but how might a hydrophilic signaling molecule behave? Yes. Bind to a receptor. Okay, and where is it coming from? Where? Another cell, secreted by another cell. Okay. Okay. So that's that. And then receptor on another cell. Well, that's that. Okay. That's what that is. Okay, so let's go back up here. Hydrophobic signaling molecules are molecules that diffuse off the across the plasma membrane. What do they bind to? So they go across the plasma membrane, and where do they go? Yes. Yes, receptors inside the membrane. And what did we call them when I taught that to you? Nuclear receptors. Right, so they go across the membrane to nuclear receptors. And so there, you need to stop and think about nuclear receptors. Okay, so what can you tell me about nuclear receptors? Everyone who's had recitation should really know by now. What can you tell me about them? Yes? Okay, so they're usually in a conformation that's bound to an inhibitory molecule, so you can, draw them somehow bound to some kind of inhibitory molecule. So what kind of domains do nuclear receptors have?
DNA domain.
So you'll have a DNA binding domain, a ligand binding domain, and then a transactivation domain, right? Or an activation. That's a transcriptional activation domain. Okay. Okay, so when the ligand binds, what happens? So first of all, is this active or inactive? Okay, when a ligand binds, what happens to this receptor? I'm hearing conformational change. What else? Inhibitory protein is released. What else? What's exposed?
The DNA binding.
DNA binding domain. Where does it go?
The nucleus.
To the nucleus, right? So let's do ligand. And then you're going to change your conformation. So we'll have ligand bound here. DNA binding domain is available. And then go into nucleus. I feel like the nucleus should be all the way over here. To nucleus. Okay. So in the, yeah.
What is the activation domain?
Activation domain, so that's a good question. So now it's in the nucleus, and there's still this activation domain here. So the DNA binding domain is going to bind to what inside the nucleus?
Promoter.
A promoter, an enhancer, some kind of DNA element that regulates transcription. And then what's the purpose of that? Let's say you're going to activate transcription. What else do you need to recruit? Transcription factors or coactivators. And so they might combine. So this would be where your coactivator combined in that activation domain. That's probably more apt to happen in the nucleus, but whatever.
Okay, that's where like transcription factors.
Yeah, so then it's going to go into the nucleus, and then it's going to come in contact with the DNA. I'm not drawing a helix. And then you're going to have your promoter and some enhancer regions. And then, so it's going to come and bind here. So here will be your newly conformed, here's your DNA binding domain, here's your nuclear receptor. And it's going to bind to some activation, some co-activator. And what's it going to recruit? What? I heard polymerase, but I think I heard the wrong polymerase. RNA polymerase, right. So then it's going to recruit RNA Pol. And once RNA polymerase is recruited, you can get gene expression, so transcription. Okay? Did I leave anything out about nuclear receptors and hydrophobic signaling? Yeah.
So, in the line imbalance, I suppose, is the NLS,
Yes, NOS. I actually looked up because someone came and visited me today and asked me specifically about that. And it just so happens the NOS tends to be near the DNA binding domain, so it's convenient that that's obscured in the absence of ligand by binding of the inhibitor. Yes, thank you. There's an NOS exposed. Yeah.
What is NO?
Nitric oxide. We spoke about that in Viagra briefly. Yeah.
So when it binds to the enhancer, it recruits what?
Co-activators, RNA polymerase, activate gene expression. Yeah. So you could say that a nuclear receptor is kind of a receptor and a transcription factor in one protein, right? Yeah. Transcription. Yeah. Sorry. TX for me is always transcription. It can be treatment too if you're a doctor. Okay, so did I leave anything out? Any questions about nuclear receptors or any of this stuff while we're on this? Because I'm going to move on to the next one.
Does that say co-activator there? What's the ligand is second one? Does that say
Here,
ligand?
co-activator.
This co-activator.
Yep. Okay, so I'm going to, yep.
The nuclear receptor, is that like a domain? Like what is that?
Nuclear receptor is a protein that has different domains with different functions, and based on its conformation and the proteins that it's associated with will either be inactive in the cytoplasm or active in the nucleus and binding to DNA and recruiting.
Also
That whole protein is a nuclear receptor. Yeah, so we only spent a few minutes on it in lecture. Yeah.
If the DNA domain is binding to the promoter or the enhancer?
It's probably the enhancer combined anywhere in the DNA in a place that think of it as binding to a DNA element that's involved with activation of gene
So they
expression.
can bind at technically both. Or any
Call
one-
it an enhancer. Let's call it
or
an
an
enhancer.
enhancer.
Because your RNA polymerase is going to be recruited. Yeah. Yes.
co-activators aren't always required, right?
Co-activators are not always required. No, they're I guess not, but often with gene expression, many transcription factors are involved, and often co-activators have to bring in other things that are required for the end goal being recruitment of RNA polymerase. Okay. Good. Are we happy? Okay, so we're going to move on. So that was that. Oh, I should have moved higher. Okay, so we're going to go like. Okay. Hydrophilic. Okay. So they bind to receptors, secreted by cells. Okay. Yes.
Well, is the transcription factor the co-activator?
Transcription factors, any protein that's going to be involved in regulation of transcription. So the nuclear receptor itself is a transcription factor, as is the co-activator in a way. Yeah. Okay.
How does the nuclear receptor interact with coactivator?
How does the nuclear receptor interact with the co-activator? When the ligand binds, the conformation change is such that it recruits the co-activator. Okay. I wish I had more chalk. Okay, so hydrophilic signaling molecules bind to receptors. What kinds of receptors did we learn about that we spent a lot of time on? GPCRs. And so we're going to do these two hydrophilic signaling molecules and receptor on cells together because they're kind of going to be in the same category. GPCRs. So let's start with GPCRs. Oh, actually, no. GPCRs, what's the other one? Kinase associated, or how about enzyme coupled receptors? Because they either have intrinsic or associated, right? So okay, so enzyme coupled. Sorry, you guys can all come over here after and take pictures with your phones. Enzyme coupled could be two categories, right? God, I'm going to be scratching my nails on the board shortly. What's one category of enzyme coupled? Broad category. Yeah. Enzyme linked. Okay. So enzyme linked, does that mean it has intrinsic activity? Right. So enzyme linked, or we'll just say intrinsic activity so there's no confusion. Intrinsic kinase activity. And then the other one is what? Associated, right? Kinase. Okay. And so then GPRs, there were also two kinds, right? Maybe based on the G proteins, right? There were inhibitory, well, there were multiple kinds, but we spoke about inhibitory, where it was the G sub I, and stimulatory, G sub S. So you can have different G proteins. You can have inhibitory and stimulatory, right? Okay. So let's move forward with GPCRs. So how do GPCRs work? What are the components? Let's start very basic. What are the components of a GPCR? G protein, right? So you have your G protein, which could be inhibitory or stimulating, and then you have your actual receptor, right? And then what else? And so that's your GPCR, but what's the purpose of the GPCR? What does it do? You've got your receptor that binds to what? What binds to a receptor? A ligand, right? Ligand binds to receptor, conformational change in receptor, which does what to the G protein? Huh? Okay, so it recruits the G protein and then does what? Not phosphorylates it. What did I hear? Activates it. Okay, so you get, so we got ligand. Oh my God, ligand. I can't write. See, you know what you guys will like say, I can't listen and write. Try talking and writing. Okay. Okay. Ligand, receptor, G protein. What happens to the G protein now? It gets activated, but what happens really? Yes. Right. So that ligand binding to the receptor results in a conformational change in your receptor, which results in a conformational change in the G protein such that GDP is released. And there's a high concentration of GTP in the cell, so GTP binds. Okay? So ligand binds receptor, binds G protein, and then GDP goes away, and then GTP binds. So now it's active. Okay, and what does that mean? So what's the point of activating the G protein? What does it do then? Does anyone remember? This is where we started talking about second messengers, these small intracellular signaling molecules. Yeah. So it activates adenyl cyclase to make cyclic AMP. That was one example. So let's go with that example. I think I gave you 2 examples, but let's go with that one because that's a nice complete one. Right? So that activates. So it turns on. We'll just do an up arrow for on. And so if you ever, someone was asking me about adenyl cyclase the other day, and I looked up the mechanism, and I don't know, but so if you ever are wondering what adenyl cyclase does, you can actually realize it's like in the word, right? It takes AMP and cyclizes it, so it makes it cyclic. But anyways, cyclic AMP. So adenyl cyclase makes cyclic AMP, right? What's the purpose of saying that cyclic AMP everywhere, and what does that do? Activates PKA, right? Because remember, PKA was bound to an inhibitor, and when the cyclic AMP binds, it releases the PKA to activate it. So, PKA. And what's the point of activating PKA? What is it phosphorylate? Transcription factor. So PKA does 2 things, right? It goes into the nucleus and phosphorylates transcription factors. And then you get gene expression. Or regulation. I don't want to always make the assumption that the gene is being turned on. It's regulation of gene expression. There might be things being turned off sometimes. What else does PKA do? It also phosphorylates the GPCR. So then PKA, negative feedback. Phosphorylates the GPCR. Okay, so that's one nice example. Yes?
Why is that negative feedback?
Negative feedback, or feedback inhibition. So whenever you turn something on that then turns itself off, you can think of that as negative feedback or feedback inhibition.
Yeah.
So you've turned on PKA. You've turned on gene expression. But then it goes and it phosphorylates your GPCR. What does phosphorylation of your GPCR do? So I heard endocytosis. How does it endocytose the receptor? Recruits arrestin. So then you get recruitment of arrestin to the phosphorylated receptor. And then the, yeah. So negative feedback, phosphorylation of GPCR. There's 2 things. One is endocytosis via arrestin. And the other is what else? So that cytoplasmic domain of the GPCR needs to interact with the G protein. So it also prevents interaction with G protein, which also shuts off signaling. Okay. Did I leave anything out? I'm sure I did. Yes?
So is it all right to generalize that whenever like GPCRs are phosphorylated, arrestin is one that's going to recruit arrestin?
I think tend to generalize, and it's very dangerous to generalize. What I taught you is that phosphorylation of the cytoplasmic domain of GPCRs is a mechanism for shutoff. So I think that's okay to assume. I wouldn't go so far with the enzyme-coupled receptors because that's a different story. Okay? Do you get phosphorylation and activation of GPCRs? Maybe, but I haven't taught you that. So I'm not really sure. It might be out there. There's probably papers. Yes?
You said this was an example of one of the things that was
Oh, I have no idea. What did I say? This is just one example of GPCRs. So sometimes it's very nice to organize things this way for me. I don't know if it's helping. Okay, so okay, so let's go down to enzyme coupled. Okay? So intrinsic kinase activity, what does that mean? Okay, so what has its own kinase domain? The receptor, right? So the receptor cytoplasmic tail has its own kinase domain, which means it can phosphorylate things. Okay, good. So did I give you yet? Did I give you? Well, so let's wait. Let's wait for. Well, no, we'll categorize some pathways right now. So did I give you any examples of? Did I give you examples of pathways with intrinsic kinase activity? Well, like specific pathways.
No.
Specific pathways. So what pathways we do? We did Wnt. That's a GPCR pathway, isn't it? NF-kappa B pathway. JAK-STAT is kinase associated. So let's put that right there. So an example of kinase associated is JAK-STAT. I'm feeling the need to look a few things up. Let me look real quick.